All right, hello everyone. Today we are going to put on our detective hats and step into the world of epidemiology. We're going to unpack how scientists track, understand, and ultimately control the spread of infectious diseases. And we'll do it by following the molecular clues left behind by a particularly deadly filovirus.
So, here's our game plan. We'll start with a pretty terrifying mystery that unfolded in the Democratic Republic of Congo. Then, we'll see how genetic sequencing completely cracked the case wide open. After that, we're going to get to grips with R naught, which is, probably the most important single number in all of epidemiology. From there, we'll look at the actual playbook for controlling an epidemic. And finally, we'll get into why the social networks we're all a part of are so critical for disease modeling today. Let's get into it. Okay, so our story kicks off in October 1998. Word starts spreading from a village called Durba in the DRC, and the news is not good. There's a mysterious and absolutely terrifying illness. People are suffering from a severe hemorrhagic fever, and at this point, nobody has a clue what's causing it. And this is what it looked like on the ground. You've got an international team of experts. They're kitted out in full hazmat gear, and they're dealing with what they know is a highly dangerous, totally unknown pathogen. You can just imagine how tense the situation was. The stakes, well, they couldn't have been higher. It actually took a full 7 months, all the way until May of 1999, for them to finally identify the culprit. And under the electron microscope, it looked like this. This is the Marburg virus, a very nasty relative of Ebola. They had their pathogen. Right, so they've identified the pathogen. But as you all know, that's really only half the battle. Knowing what it is one thing. The really crucial questions are, how is it spreading? And most importantly, where is it coming from? The first massive clue came from looking at this epicurve. Now, if you look at these charts, you'll see something a bit odd. Instead of one big, explosive outbreak that peaks and then fades, the cases are just sort of popping up sporadically over 2 whole years. And that bottom chart is really telling. Look at how the vast majority of cases were among local gold miners. This sustained, low-level pattern was really unusual. So this strange pattern led the investigators to ask a really central question. Were they looking at one single, long, slow-burning chain of people infecting other people? Or was something else going on here entirely? And to figure that out, they went straight to the virus's genetic code. It all boiled down to 2 competing hypotheses. On one hand, if it's just one big chain of person-to-person spread, the virus's genome just doesn't have time to change very much. All the sequences you collect from patients should be really similar, almost identical. But if people are getting infected over and over again from some natural source, say an animal reservoir, you would expect to see way more genetic diversity in your samples. And boom, this phylogenetic tree was the smoking gun. As you guys know, the branch links here show genetic distance, and what they found was just incredible. Instead of all the samples clustering together, they saw multiple really distinct genetic lineages. You can see at least 9 of them branching off here. This massive genetic divergence completely blew that single chain idea out of the water. This wasn't one outbreak, it was lots of little ones, repeatedly spilling over from whatever its natural home was.
So here's the key takeaway. Marburg is, without a doubt, a terrifying virus. But what the data showed was that it wasn't actually very good at spreading between people. Most infections were just dead ends. In fact, the longest human-to-human chain they could find was only 7 people long. And that led to the conclusion that we humans are what's called spillover hosts. We can get sick, sure, but we aren't the natural reservoir where the virus hangs out and persists. So this brings us to a really fundamental question. We figured out that Marburg wasn't spreading all that well between people, but how do we actually quantify that? How do you put a hard number on a pathogen's ability to spread?
The answer lies in this absolutely fundamental metric, the basic reproduction number, which you'll know as R naught or R0. It's a theoretical number, and it tells you, on average, how many other people one single sick person is going to infect if you drop them into a population where absolutely no one has any immunity. It's the pathogen's raw potential.
And the value of R naught tells you pretty much everything you need to know about where an outbreak is headed. If it's greater than 1, each case is creating more than one new case, and your epidemic is going to grow, probably exponentially. If it's less than 1, the outbreak is going to fizzle out all on its own. It's a dead end. And for Marburg, in that Durba outbreak, the R naught for human-to-human spread was clearly, thankfully, less than 1. And just to put that into perspective, have a look at some of these numbers. Measles is an absolute beast, a transmission machine, with an R naught somewhere between 12 and 18. Think about that. one person with measles can infect an entire classroom. It just really hammers home how poorly adapted Marburg was for spreading in people at that point in time. So, what do we do with this knowledge? How does it translate into action? Well, R naught isn't just some academic number you calculate after the fact. For anyone in public health, it is the foundation of the entire playbook for how you control a disease.
Okay, so the value of R naught comes from this simple but really powerful formula. It's a product of three things. First, you've got C, the contact rate. Then you've got rho, the Greek letter, which is the probability of transmission happening during one of those contacts. And finally, you've got gamma, which is just the duration of infectiousness. To control an outbreak, you have to attack these variables. Each one is a lever that we can pull. And this is literally how public health interventions work. You want to reduce C, the number of contacts. You use things like quarantine or social distancing. You want to reduce rho, the probability of transmission. That's where masks, hand washing, and PPE come in. And you want to reduce gamma, how long someone is infectious. You use antiviral drugs to help them clear the virus faster. Every single strategy is designed to manipulate this equation and, crucially, to drive that R number below 1.
And this graph shows that concept in action perfectly. See, R naught is the raw, theoretical potential of a pathogen in a perfect world. But what health officials are tracking day to day is the effective R number, sometimes called RT. This is what's happening in real time, in a real population that has some immunity and has all these control measures going on. You can see right here how it was pushed above and below that critical line of 1 during the COVID-19 pandemic in Scotland. But to build really good, sophisticated models, we have to go even deeper. Thinking of that contact rate C as just one average number for a whole population is a helpful starting point, but the reality is just so much more complex. For instance, a big debate in epidemiology is whether transmission is density-dependent or frequency-dependent. So for something like the flu, you might assume it's density-dependent, right? The more crowded a city is, the more contacts people have, the faster it spreads. Makes sense. But for, say, a sexually transmitted infection, the number of partners a person has might not really change whether they live in a tiny village or a huge city. That's frequency dependence. And the kind of model you build is completely different depending on which one of those assumptions you make.
And really, this all leads us to the absolute frontier of modern epidemiology. It's not just about how many people you bump into. It's about who they are. It's about how our friend groups and communities are structured and how our global travel networks connect every single one of us. Understanding these incredibly intricate networks, that's the real key to predicting and hopefully preventing the next big pandemic. Why does one outbreak fizzle out while another one goes global? The answer is hiding right there in the patterns of our connections.